Biotechnology, Chapter 9, Biotechnology Principles and Processes. Ever since the days of René Descartes, the French philosopher, mathematician and biologist of 17th century, all humans' knowledge, especially natural science, were directed to develop technologies which aid to create creature comfort of humans' lives, age also value of human lives. The whole approach to understanding natural phenomena become anthropocentric. Physics and chemistry gave rise to engineering, technologies, and industries which all worked for human comfort and welfare. The major utility of biological world is as source of food. Biotechnology, the 20th century offshoot of modern biology, changed our daily life as its product brought qualitative improvement in health and food production. The basic principle of underlying biotechnology process and some application are highlighted and discussed in this unit. Herbert Boyer, 1936. Herbert Boyer was born in 1936 and brought up to corner western peninsula, where railroads and mines were destiny of most young men. He completed graduate work at University of Pittsburgh in 1963, followed by 3 years of postgraduate study at Yale. In 1966, Boyer took over assistant professorship at the University of California at San Francisco. By 1969, he performed studies on the couple of restriction enzyme of E. coli bacterium with especially useful properties. Boyer observed that these enzymes have the capability of cutting DNA strands in particular fashions, which left what has become known as sticky ends on the strands. These clip ends made pasting together piece of DNA as precise exercise. This discovery in turn led to a rich and rewarding conversation in Hawaii with a Stanford scientist named Stanley Cohen. Cohen had been studying small ringlet of DNA called plasmids and which float about freely in cytoplasm of certain bacterial cell and replicate independently from coding strand of DNA. Cohen had developed a method of removing these plasmids from the cell and these reinserting them in other cell. Combining this process with that of DNA splicing enabled Boyer and Cohen to recombine segment of DNA in desired configuration and insert the DNA in bacterial cell, which could then act as manufacturing plant for a specific protein. This breakthrough was the basis upon which the discipline of biotechnology was founded. Chapter 9, Biotechnology, Principles and Processes. 9.1, Principles of Biotechnology. 9.2, Tools of Recombinant DNA Technology. 9.3, Processes of Recombinant DNA Technology. Biotechnology deals with technique of using live organism and enzyme from organism to produce product and process useful to humans. In this sense, making the curd, bread and wine, which are all microbes mediated process, could also be thought as a form of biotechnology. However, it is used in restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on a larger scale. Further, many other process techniques are also included under biotechnology. For example, in vitro fertilization leading to the test tube baby, synthesizing a gene and using it, developing a DNA vaccine or correcting a defective gene are all part of biotechnology. The European Federation of Biotechnology (EFB) has given a definition of biotechnology that encompasses both a traditional view and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural science and organism cells, part thereof, and molecular analogs for product and service. 9.1 Principle of biotechnology. Among many, the two core techniques that enabled birth of modern biotechnology. First, genetic engineering technique to alter the chemistry of genetic material DNA or RNA, asked in NEET 1996. To introduce these into host organism and thus change the phenotype of host organism. Second, bioprocesses engineering. Maintenance of sterile microbial contamination free ambience in chemical engineering process to enable growth of only the desired microeukaryotic cell in large quantity for manufacture of biotechnological product like antibiotics, vaccine, enzyme, etc. Let us now understand the conceptual development of principle of genetic engineering. You probably appreciate the advantage of sexual reproduction over asexual reproduction. The former provides the opportunity of variation and formulation of unique combination of genetic set of some of which may be beneficial to organism as well as population. Asexual reproduction preserves the genetic information while sexual reproduction permits variation. Traditional hybridization procedure used in plant and animal breeding very often led to inclusion of multiplication of undesirable gene along with desired gene. The technique of genetic engineering, which include creation of recombinant DNA used for gene cloning and gene transfer, overcome this limitation and allow us to isolate introducing only one set of desirable gene without introducing undesirable genes into targeted organism, asked in NEET 2011. Do you know like fate of piece of DNA which is somehow transferred into an alien organism? Most likely the piece of DNA would not be able to multiply itself in progeny cell of the organism, but when it gets integrated into genome of recipient, it may multiply and be inherited along with the host of DNA. This is because the alien piece of DNA has become part of chromosomes, which has ability to replicate. In a chromosome, there is a specific DNA sequence called origin of replication, which is responsible for initiating replication. Therefore, for the multiplication of any alien piece of DNA in which an organism, it needs to be part of chromosomes, which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in host organism. This can also be called as cloning or making multiple identical copy of any template DNA. Let us now focus on first instance of construction of an artificial recombinant DNA molecule. The construction of first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid, autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA at a specific location became possible with the discovery of so-called molecular seizure restriction enzyme, asked in 2005. The cut piece of DNA was then linked with plasmid DNA. This plasmid DNA acts as vector to transfer the piece of DNA attached to it. You probably know that mosquito act as insect vector to transfer malarial parasite into human body. In this way, a plasmid can be used to vector deliver alien species of DNA into host organism. The linking of antibiotic resistance gene with plasmid vector become possible with enzyme DNA ligase, which act as cut part of join their ends. This make a new combination of circular autosomally replicating DNA create in vitro known as recombinant DNA. When this DNA transferred into Escherichia coli, A bacterium closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene in Escherichia coli. You can hence interfere here three basic steps of genetic modify organism. First, identification of undesirable gene. Second, introduction of identified DNA into host. Third, maintenance of introduction of DNA host transferred into DNA of progeny. 9.2 Tools of recombinant DNA technology. Now we know the foregoing discussion of that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tool, that is restriction enzyme, polymerase enzyme, ligase vector and host organism. Let us try some of these in detail. 9.1.2.1 Restriction enzyme. In year 1963, two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added to methyl group of DNA while other cut the DNA. The latter was called restriction endonuclease. The first restriction endonuclease hence second, whose functioning depend on a specific DNA nucleotide sequence, was isolated and characterized by 5 years later. It was found that hence second always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. 6 base pair recognized. This specific base sequence is known as the recognition sequence for hence second. Besides hence second, today we know more than 900 restriction enzymes that have been isolated over 230 strains of bacteria, each which recognize different recognition sequence. The convention of naming these enzymes in first letter of the name comes from genus, and second two letter come from species of prokaryote from which they are isolated. EcoR1 comes from Escherichia coli RY13. In EcoR1, the letter R derived from the name of strain. Roman number of the following the names are indicated in order by the enzymes were isolated from the strain of bacteria. Restriction enzyme belong to the larger class of enzyme called nucleases. These are two kind exonucleases and endonucleases. Asked in 2020. Exonucleases remove nucleotide from the ends of the DNA, whereas endonucleases make cut at a specific position within DNA. Each restriction endonuclease function by the inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to DNA and cut each of two strands of double helix specific point in sugar phosphate backbone. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in DNA. Action of restriction enzyme. The enzyme cut DNA at a same strand. EcoR1 cut DNA between the base pair G and A only when sequence GAATTC are present in DNA. Vector DNA, foreign DNA. DNA fragment joined at sticky ends. This is sticky ends. AATTA. Recombinant DNA. Step of formation of recombinant DNA by action of restriction endonuclease by EcoR1. Do you know what palindromes are? These are a group of letters that form the same word when read from both backward and forward. Malayalam. As against the word palindrome, where the same word is read both directions. The palindrome in DNA is a sequence of base pairs that read same on two strands when orientation of reading is kept same. For example, the following sequence reads in two strands 5' to 3' direction, also to 3' to 5'. This is palindromic sequence. GAA-TTC. Restriction enzyme cuts the DNA strand little away from the center of palindrome site, but between the same two bases on opposite strand. This leaves single stranded portion at the ends. There are overhanging strands called streaky ends on each strand. These are named so because they form hydrogen bond with their complementary cut counterpart. This stickiness of end facilitates the action of enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant DNA, which are composed of DNA from different sources of genome. When cut by same restriction enzyme, the DNA fragment have same kind of sticky end, and these can be joined together end to end using DNA ligase. Foreign DNA, vector plasmid. Same restriction enzyme cut both ends of DNA and vector at a specific point. Ligase join foreign DNA and plasmid. Recombinant DNA molecule transformation. E. coli cloning host cell divide. Diagrammatic representation of recombinant DNA technology. This is. You may realize normally unless cut vector and DNA source DNA with same restriction enzyme, the recombinant vector molecule cannot be created. Separation and isolation of DNA fragment. The cutting of DNA by restriction endonuclease result in fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to remove toward the anode under an electric field through a medium matrix. Nowadays, the most commonly used matrix is agarose, which is natural polymer extracted from seaweeds. The DNA fragments separated and resolved according to their size through shaping effect provided by agarose gel. Hence, the smaller the fragment size, the farther it moves. Look in figure 9.3 and guess in which gel sample was loaded. The separated DNA fragment can be visualized staining with DNA with compound known as ethidium bromide followed by exposure of UV radiation. You can not see pure DNA fragment. DNA fragments are visible. DNA fragment in the visible light and without staining. You can see bright orange color of bands of DNA in ethidium bromide stained with the stained gel exposed to UV light. The separated bands of DNA are cut out from the agarose gel and extracted from gel piece. This step is known as elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vector. A typical agarose gel electrophoresis showing migration of undigested lane 1, undigested lane and digested set 2 to 4 digested lane. Cloning vector 9.2.2 cloning vector. You know that plasmid and bacteriophage have ability to replicate within bacterial cell independent to control of chromosomes of DNA. Bacteriophage become because of their high number of per cell have very high copy number and their genome within bacterial cell. Some plasmids may have only 1 or 2 copies per cell, whereas other may have 15 to 100 copies per cell. The number can go even higher. If we are able to link an alien piece of DNA with bacteriophage of plasmid DNA, we can multiply its number equal to the copy number of plasmid or bacteriophage. Vector used in present engineered in such a way that they help easy linking of foreign DNA and selection of recombinant from non-recombinants. The following are features required for facilitated cloning into vector. First, origin of replication. ORI. This is a sequence from where replication starts, and any piece of DNA when linked to this sequence can be made to replicate within the host. This sequence is also responsible for controlling copy number of linked DNA. So, if one want to recover many copy of target DNA, it should be cloned in a vector whose origin support high copy number. Second, selectable markers. In addition to origin of replication, vector require a selectable marker which help in identify the eliminating non-transformants, selectively permitting the growth of the transformant. Transformation is a procedure through which a piece of DNA is introduced into host bacterium. You will study the process subsequent section. Normally, the genes encoding resistance to antibiotics such as ampicillin, chlorophenicol, and tetracycline or kanamycin are etc. considered useful selectable marker for E. coli. The normal E. coli cell do not carry resistance against any of these antibiotics. Third, cloning site. In order to link the alien DNA, the vector needs to have very few, preferably single recognition sites for commonly used restriction enzyme. Presence of more than one recognition site within the vector will generate several fragments, which will complicate the gene cloning. The ligation of DNA is carried out at the restriction site present in two antibiotic resistant genes. For example, you can ligate the foreign DNA at BAMH1 site of tetracycline resistance gene in vector PBR322. The recombinant plasmid will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on tetracycline containing medium. The transformant growing on an ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinant will grow in ampicillin containing medium but not on containing tetracycline. But non-recombinant will grow on the medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transformant, whereas other antibiotic resistance gene get inactivated due to insertion of alien DNA and help in selection of recombinant. Here, figure 9.4. Escherichia coli cloning vector PBR322 restriction site hinge third, eco R1, BAMH1, cell 1, PVU second, PhD first, CLAF CLA first, ORI and antibiotic resistance gene, ampicillin, tetracycline, ROP codes for the protein involved in replication of plasmid. BAMH1 tetracycline, BAMH1 cell, ROP, PVU second, origin of ORI, ampicillin, PST 1, PVU first, CLA first, hint third, eco R1. Selection of recombinant due to inactivation of recombinant is a cumbersome procedure because it requires simultaneous plating of two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinant from non-recombinant on the basis of their ability to produce color in presence of chromogenic substrate. In this recombinant DNA, the DNA is inserted within the coding sequence of an enzyme, beta-galactosidase. This results into inactivation of the gene for synthesis of enzyme, which is referred to insertional inactivation. The process of chromogenic substrate gives blue color to clonage if the plasmid in bacteria does not have an insert. Presence of insert result into insertional inactivation of the beta-galactosidase gene and colonies do not produce any color. These are identified as recombinant colonies. 4. Vector for cloning gene in plant and animals. You may be surprised to know that we have learned the lesson of transferring genes into plant and animals from bacteria and viruses which have known this for ages. How to deliver genes to transform eukaryotic cell and force them to do what the bacteria or viruses want. For example, Agrobacterium tumefacium, a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA to transform normal plant cell into tumor and direct these tumor cells to produce the chemical required by pathogens. Similarly, retrovirus in animals have ability to transform normal cells into cancerous cells. A better understanding of art delivering genes by pathogens in their eukaryotic host has generated knowledge to transform these tools of pathogen into useful vector for delivering genes of insert to humans. The tumor-inducing Ti plasmid of Agrobacterium tumefacium has now been modified into cloning vector, which is no more pathogenic to the plant but is still able to use mechanism to deliver genes of our interest into a variety of plants. Similarly, retrovirus have also been disarmed and are now used to deliver desirable genes into animal cells. So, once a gene of DNA fragment has been ligated into suitable vector, it is transferred into bacterial, plant or animal host where it multiplies. 9.2.3 Competent host for transformation with recombinant DNA. Since DNA is hydrophilic molecule, it cannot pass through cell's membrane. Why? In order to force bacteria to take up the plasmid, the cell must be first made competent to take up DNA. This is done by treating them with sequence concentration of divalent cation such as calcium, which increase the efficiency with which DNA enters in the bacterium through pores in its cell. Recombinant DNA can then be forced into such cell by incubating the cells with recombinant DNA on ice, followed by the placing briefly at 42 degrees Celsius heat shock, and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. This is not only a way to introduce alien piece of DNA to host cell. In a method known as microinjection, recombinant DNA is directly injected into nucleus of animal cell. In another method suitable for plants, cells are bombarded with high velocity of microparticles gold tungsten coated with DNA and method known as bioelastic or gene gun. And the last method used for disarmed pathogen vector, which when allowed to infect the cell transfer recombinant DNA into host. Now that we have learned about tools in constructing recombinant DNA, let us discuss the process facilitating the recombinant DNA technology. 9.3 Process of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence such as isolation of DNA, fragmentation of DNA by restriction into nucleases, isolation of desired DNA fragments, ligation of DNA fragment into vector, transferring the recombinant DNA into host, culturing the host cell in the medium at large scale, extraction of desirable product. Let us examine each of these steps in some details. 9.3.1 Isolation of genetic material DNA. Recall that nucleic acid is genetic material of all organisms without exception. In majority of organisms, this is deoxyribonucleic acid DNA. In order to cut the DNA with restriction enzyme, it need to be pure form, free from other macromolecules. Since the DNA is enclosed within the membrane, we have to break the cell open to release DNA along with macromolecules such as RNA, protein, polysaccharide and also lipid. This can be also achieved by treating the bacterial cell, plant and animal tissue with enzyme, lysozyme in bacteria, cellulose in plant cell, like chitinase in fungus. You know that genes are located along the molecules of DNA intertwined with proteins such as histone. The DNA can be removed by treatment with nuclease, whereas protein can be removed by treatment with protease. Other molecules can be removed by appropriate treatment with and purified DNA ultimately precipitates out and the addition of chilled ethanol. This can be seen in collection of fine thread in the suspension. DNA separated out can be removed by spooling. 9.3.2 Cutting of DNA at a specific location. Restriction enzyme digestion are performed by incubating purified DNA molecule within restriction enzyme. At the optimal condition of that specific enzyme, a gross gel electrophoresis is employed to check the progression of restriction enzyme digestion. DNA is a negatively charged molecule, hence it moves toward positive electrode anode. The process repeat with vector DNA also. The joining of DNA involves several processes after having cut the source of DNA as well as the vector DNA with a specific restriction enzyme. The cut out gene of interest from source of DNA and cut vector with space are mixed and ligase is added. This results in preparation of recombinant DNA. 9.3.3 Amplification of gene of interest using PCR. PCR is known as polymerase chain reaction. In this process, multiple copy of gene or DNA of interest is synthesized in vitro using two set of primers. Small chemically synthesized oligonucleotides and are complementary to region of DNA and the enzyme DNA polymerase. The enzyme extends the primer using nucleotide provided in the reaction and genomic DNA as template. If the process of replication of DNA repeated many times, the segment of DNA amplified approximately billion times. 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase isolated from bacterium Thermus aquaticus, which remain active during high temperature induced denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector to further cloning. Polymerase chain reaction (PCR). Each cycle has three steps: denaturation, primer annealing, extension of primer. Denaturation, annealing, extension. Region to be amplified 3' to 5', double-stranded DNA, heat 3' to 5', primer. This is DNA polymerase or Taq DNA polymerase plus deoxyribonucleic acid after third recycle, amplified 1 billion times. 9.3.4 Insertion of recombinant DNA into host cell or organism. There are several methods of introducing the ligated DNA into recipient cell. Recipient cell, after making them competent to receive, take up DNA present in its surrounding. So if the recombinant DNA bearing gene of resistance to the antibiotics as ampicillin is transferred into Escherichia coli, the host cell become transferred into ampicillin resistant cell. If we spread the transformed cell on agar plates containing ampicillin, only transformant will grow. Untransformed recipient cell will die. Since due to ampicillin resistance gene, one is able to select transformed cell in the presence of ampicillin. The ampicillin resistant gene in this case is called a selectable marker. 9.3.5 Obtaining the foreign gene product. When you insert a piece of alien DNA into cloning vector and transfer it into bacteria, plant or animal cell, the alien DNA get multiplied. In almost all recombinant DNA technology, the ultimate aim is to produce a desirable protein. Hence, there is need for recombinant DNA to be expressed. In foreign gene get expressed under appropriate condition. The expression of foreign gene in host cell involve understanding many technical details. After having cloned the gene of interest and having optimized the condition in, to induce expression of target protein, one has to consider producing it on large scale. Can you think of any reason why there is need for large scale production? If any protein encoding gene is expressed in a heterologous host, it, it is called recombinant protein. The cell harboring cloned gene of interest may be grown on a small scale in laboratory. The culture may be used for extracting the desired protein and then purifying it by using different separation techniques. The cell can also be multiplied by continuous culture system wherein used in medium drained out from one side while fresh medium is added on other side of the cell and in their physiological medium of the host. Active log exponential phase. Cell physiological most active log exponential. This type of culturing method produce a larger biomass leading to higher yields of desirable proteins. Small volume cultures cannot yield appreciable quantities of product. To produce a large quantity, the development of bioreactor where larger volume 100 to 1000 liter of culture can be processed was required. Thus, bioreactors can be thought of as vessel in which raw material are biologically converted into a specific product. Individual enzyme etc. using microbial plant, animals of human cells. A bioreactor provides the optimal condition for achieving the desired product by providing the optimal growth of condition pH, temperature, pH, substrate, salt, vitamin, oxygen. The most commonly used bioreactor are stirring type, which are shown in figure 9.7. Simple stirred tank bioreactor, simple tank bioreactor, sparge tank bioreactor through which sterile air bubble is sparged. Gas entainment, increase surface area of oxygen surface. Bubbles dramatically increase the oxygen area. Simple stirred bioreactor, sterile air, culture broth, flat bladed impeller, foam breaker, motor, acid base for pH control, steam for sterilization. A stirred tank reactor is usually cylindrical or with a curved base to facilitate the mixing of reactor contents. The stir facilitates even mixing of oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look at the finger closely, you will see that bioreactor has an agitator system, an oxygen delivery system, and foam control system, a temperature control system, pH control system, and sampling port so that a small volume of culture can be withdrawn. A bio oxygen delivery system, foam control system, temperature control system, pH control system, and sampling port. 9.3.6 Downstream processing. After completing the biosynthetic stage, the product has been subjected to through a series of processes before it is ready to marketing and as a finished product. The process includes separation and purification, in which collectively referred to as downstream processing. The product has to be formulated with suitable preservatives. Such formulation has to undergo through clinical trials, as in case of drug, strict quality control testing for each product is required. The downstream processing and quality control testing vary from product to product. Summary: Biotechnology deal with large scale production of marketing product and processes using live organism. Sale of enzyme. Modern biotechnology using genetic modified organism was made possible only when man learned to alter the chemistry of DNA and construct the recombinant DNA. This key process is called recombinant DNA technology or genetic engineering. This process involves the use of restriction endonucleases, DNA ligases, appropriate plasmid or viral vector to isolate the free of foreign DNA into host organism, expression of foreign gene, purification or gene product. The function protein is finally making a suitable formulation marketing. Large scale production involve use of bioreactor. ये तो रीडिंग हुआ, अब जा रहे हैं देखने जो क्वेश्चंस आ चुका है। जैसे कि 1994 में एक क्वेश्चन आया यहाँ से, दिस की प्रोसेस called recombinant DNA technology genetic engineering. The process involves control of restriction endonucleases. Large scale production involves bioreactor. The process includes separation and purification, which are collectively referred as downstream processing.